Imagine you are trying to pull out this really stubborn weed from your garden.
Oh, I hate those.
Right. So you get a good grip, you yank, and you pull off the top. And visually, the surface looks completely clear. You think you've won.
But the root is still there.
Exactly. The root remains. But it's actually worse than just remaining. Imagine this root has built its own secret underground bunker. And down there, it's actively manufacturing this highly specialized biochemical armor.
Wow.
Yeah, it basically renders itself completely impervious to whatever weed killer you just sprayed on top of it.
That is, that's a phenomenal way to visualize the exact clinical reality we face with acute myeloid leukemia, or AML.
Which is what we are diving into today.
Yeah. It's a notoriously aggressive blood cancer, and it's characterized by these devastatingly high relapse rates.
And those relapses, they come down to the roots, right?
Precisely. They come down to leukemic stem cells, or LSCs. They are the roots in your garden analogy. You can clear the bulk blast cells from the bloodstream.
Which makes it look like remission.
Right. It looks like a win. But if those LSCs are just, hiding out in the bone marrow microenvironment, the disease will inevitably return.
And that brings us to the actual mission for today's deep dive. We're digging into this groundbreaking 2026 study from Cell Stem Cell by Hahn and colleagues.
Such a massive paper.
It really is. It provides this exhaustive blueprint of that exact underground bunker. It's a mechanism they call the ketogenesis ferroptosis axis.
It's quite a mouthful.
It is. But don't worry, we're going to break it all down. Our goal today is to examine exactly how these LSCs rewire their own metabolism to survive, and how the researchers figured out a way to dismantle this armor without, hurting the healthy cells.
Yeah, to really understand this vulnerability, we have to look at the LSC's primary survival mechanism first.
Okay, let's get into it.
So the researchers started by profiling the metabolism of these cells. And the data immediately highlighted this striking anomaly with ketogenesis.
Now, normally ketogenesis is a liver thing, right? Like if you're on a keto diet.
Exactly. In a standard physiological context, when your systemic glucose is low, your hepatic tissue, your liver takes over. It oxidizes fat to make ketone bodies.
Things like beta-hydroxybutyrate?
Right, BHB and acetoacetate, and then those are exported out to fuel the brain and other tissues.
But when I was reading the metabolomics data in this paper, the wild thing is that AML LSCs are not relying on the liver's BHB.
Not at all.
They're essentially acting like tiny independent ketone factories.
Yeah, they are biological preppers. And to prove this, the team used something called 13C palmitate isotope tracing.
Okay, let's break that down for anyone who doesn't work with mass spectrometry every day. How does that work?
It's actually pretty elegant. You take palmitate, which is a common fatty acid, and you tag it with a heavy carbon-13 isotope.
Like putting a molecular GPS tracker on the fat.
Exactly, a GPS tracker. You feed this tag fat to the cancer cells, and then you use mass spectrometry to see exactly where those heavy carbons end up inside the cell.
And the data was just totally definitive, right?
Oh, completely. The heavy carbons from that palmitate were systematically incorporated directly into intracellular BHB.
So the cancer cells are burning the fat locally.
Yes. They're using fatty acid oxidation, or FAO, to manufacture their own ketone bodies. Yep. And the wild part is they are doing it at rates comparable to a liver cell during strict ketosis.
That is insane. But, it raises a question. Is this BHB factory actually vital for the cancer cell, or is it just like a byproduct of it being a hyperactive tumor?
Right, correlation versus causation. So they tested this by shutting down the very first step of the fat-burning pathway. They used a drug called etamoxor.
Etamoxor, what does that target?
It's a small molecule inhibitor of CPT1A. CPT1A is basically the molecular gateway that lets fatty acids enter the mitochondria to be burned.
So etamoxor essentially welds the mitochondrial door shut.
That's exactly what it does. And just to be absolutely sure, they also genetically deleted the CPT1A gene in their mouse models.
And what happened?
In both cases, the influx of fat stopped, the palmitate couldn't convert into BHB, and the LSC's internal ketone levels just plummeted.
Wow, so they definitively prove the LSCs are manufacturing it themselves.
Yep. They are autonomous factories.
Okay, let's do a quick listener check-in here, just to lock in what we've covered so far. I like to do these little bullet point recaps.
I love that.
So, bullet 1, LSCs in AML use fat burning, that's FAO, to manufacture their own ketone bodies, specifically BHB.
Perfect.
And bullet 2, this autonomous ketone factory is what separates the dangerous LSCs from normal blood cells and the bulk cancer cells.
Spot on.
So here's a question for you listening to mull over. Think about a machine that generates its own fuel. If you wanted to shut that machine down permanently, where would you attack it?
Well, logically, you go after the machinery making the fuel.
Exactly. Which brings us to the next massive finding in the paper. If they are making their own BHB, what is the specific enzyme doing it?
That's where the transcriptomic profiling came in. They screened all the enzymes involved in ketone synthesis and found the master switch. It's the rate-limiting enzyme called HMGCS2.
HMGCS2, and they found this huge asymmetry here, didn't they?
A massive asymmetry. HMGCS2 was wildly overexpressed in the leukemic stem cells. But in normal, healthy hematopoietic stem cells, the HSCs, its baseline expression was almost non-existent.
Barely detectable. Which is the holy grail in oncology, right?
Oh, absolutely. Finding a target that the cancer desperately needs, but the healthy cell doesn't even use.
But, just seeing differential expression in a data set isn't really enough to base a drug on. I want to look at the receipts. How rigorous was their modeling?
Oh, it was incredibly rigorous.
Yeah.
They didn't just use one type of leukemia model. They used multiple mouse models driven by totally distinct mutations, like MLL-AF9, HOXA9-1, and HOX9-NUP98.
Okay, so making sure this isn't just a quirk of one specific mutation.
Exactly. It's a fundamental metabolic feature of the disease. But they went further. They used patient-derived xenograft, or PDX, models.
Those are crucial. Explain the PDX setup for the listeners.
So they literally took primary bone marrow cells directly from 3 individual human AML patients. They genetically silenced the HMGCS2 gene in those human cells and then transplanted them into mice.
And the result?
Without HMGCS2, the leukemia-initiating cells completely crashed, and the survival of the mice was drastically prolonged.
Wow. But wait, what about the healthy blood cells? If you delete this enzyme, doesn't it hurt normal blood production?
That is the most important part. To test that, they used a very specific mouse model called VAVCRE, HMGCS2-RF.
VAVCRE, that's a conditional knockout system, right?
Yeah, it lets you delete a specific gene only in the normal blood-forming stem cells. So they wiped out HMGCS2 in all the healthy cells and just watched them for 16 weeks. Absolutely nothing happened. Normal blood production, red blood cells, white blood cells, it was all totally fine.
That is incredible. Let me offer a visual analogy here. HMGCS2 is basically the main choke valve in the cancer cell's ketone factory. You turn it off, the cancer starves. But the healthy cells next door, they don't even notice because they don't rely on that factory at all.
That is exactly the dynamic.
Okay, time for another quick mini recap. Bullet 1. HMGCS2 is the vital master switch controlling BHB production in these LSCs.
Yep.
Bullet 2. Shutting down HMGCS2 kills both human and mouse leukemic stem cells, but completely spares normal, healthy blood cells.
Which is the dream scenario for a drug target.
It really is. So here's a question for the audience. In cancer therapy, the absolute holy grail is killing the tumor without the toxic side effects to healthy tissue. Why do you think uncovering these highly specific metabolic choke points is so difficult?
It's a great question, because metabolism is usually so intertwined.
Exactly. But let's pivot the narrative a bit, because I want to push back on something I said earlier.
Okay, go for it.
I said you turn off the factory and the cancer cell starves. Sounds super simple, right?
Right.
But that's not actually what happens.
No, it's not starvation at all. It's a totally different, highly explosive type of cell death called ferroptosis.
Ferroptosis. Okay, we need to unpack this. What exactly is ferroptosis?
So ferroptosis is an iron-dependent form of cell death. It's completely distinct from standard apoptosis, which is how cells normally program themselves to die. Ferroptosis is triggered when specific fats in the cell membrane oxidize and basically shatter the cell.
Oxidize, like rusting.
Exactly like rusting.
Yeah.
Specifically, it happens to these fats called polyunsaturated fatty acid phospholipids, or PUFAPLs for short.
PUFAPLs. Okay, let's use an image to remember this. Imagine those PUFAPL fats in the membrane are like bone-dry brush in a dense forest.
Oh, okay.
And the cancer cell, because it has this hyperactive metabolism, it's constantly throwing off sparks.
Right, reactive oxygen species, or MTROS, those are the sparks.
Exactly. So normally the BHB that the cell makes acts like a giant sprinkler system. It keeps everything wet.
Right, it protects the membrane.
But when you delete HMGCS2, you turn off the sprinkler, the sparks hit that dry PUFAPL brush, and boom, the entire cell membrane catches fire and burns down.
That is a perfect way to describe it. The membrane literally rusts and shatters from the inside out.
And the researchers actually saw this happening physically, right?
Yeah, they measured huge spikes in malonyaldehyde, or MDA, and they used BO-DIPY C11 staining. Both of those are specific markers for fats oxidizing out of control.
And didn't they use electron microscopy too?
They did. And under the EM, you don't see the normal signs of apoptosis.
Yeah.
You see mitochondria that are shrunken with completely collapsed cristae. That is the physical hallmark of ferroptosis.
But how did they prove definitively that it was ferroptosis and not just standard apoptosis?
They did rescue experiments. They took these vulnerable dying LSCs and treated them with fire extinguishers, basically ferroptosis inhibitors like leproxstatin-1, ferrostatin-1, and alpha-tocopherol.
And they survived?
They completely survived. But when they used an apoptosis inhibitor, a drug called ZVAD-FMK, it did absolutely nothing. The cells still blew up.
Wow. Okay, let's do our third recap. Bullet 1. Deleting HMGCS2 does not starve the LSC. It causes it to die via this explosive process called ferroptosis.
Right.
Bullet 2. Ferroptosis happens when specific fats in the membrane, those PUFAPLs, oxidize and destroy the structural integrity of the cell.
The dry brush catches fire.
Exactly. So for you listening, here's a thought. Knowing that cancer cells can essentially rust to death, how might this change the way we design future chemotherapy drugs?
It opens up a whole new avenue for drug design, honestly, bypassing standard apoptosis entirely.
It's fascinating, but, there's still a massive missing link here.
The mechanism.
Yeah. How exactly does a metabolic fuel like BHB act like a sprinkler system? How does it actually stop the membrane from turning into dry brush? I mean, it's not just floating around acting like a simple antioxidant, is it?
No, it's much more profound than that. The BHB is actually acting as an epigenetic regulator.
Epigenetics. Okay, so we are talking about physically changing how the DNA is read.
Exactly. The researchers did quantitative proteomic profiling, and they found that BHB physically attaches to histones.
And histones are the little protein spools that our DNA winds around.
Right. The process is called lysine beta-hydroxybutyrylation, or KBHB for short. The BHB binds to specific histones, H2B, H3, and H3K4.
So the fuel is literally modifying the packaging of the DNA.
Yes, and to figure out exactly what that modification was doing, they combined ATAC-seq with RNA-seq.
Okay, ATAC-seq is super cool. It basically maps out exactly which parts of the DNA are unspooled and physically open to being read, right?
Exactly. It shows you the accessible genome. And when they overlaid that with the RNA expression data, they found that this BHB modification tightly controls one specific gene.
Which gene?
FADS2. Fatty acid desaturase 2.
Wait, FADS2, that's the enzyme that manufactures the POAPLs, isn't it?
Yes. FADS2 is the factory that makes the dry brush.
Oh my God, the feedback loop here is insane. Let me see if I have this right. The cell burns fat to make BHB. The BHB goes to the nucleus and puts an epigenetic padlock on the DNA.
Specifically on the FADS2 gene.
Right. It locks the drawer labeled FADS2, it turns that gene down. And because FADS2 is turned down, the cell stops manufacturing the highly combustible PUFA fats.
You've got it perfectly. Less FADS2 means less combustible fat in the membrane, which makes the cell immune to the sparks it's generating. It makes it immune to ferroptosis.
The metabolism is literally rewriting the cell's genetics to build its own armor.
It is. And when the researchers used genetic tools to manually silence FADS2 in those dying, vulnerable cells, the dry brush disappeared, the lipid peroxidation stopped, and the cancer cells survived again.
That is it's breathtaking molecular biology. Okay, let's recap this final mechanism for everyone.
What's it?
Bullet 1, BHB attaches to DNA histones that KBSB tag to physically alter gene expression. That's epigenetics.
Yep.
Bullet 2, this epigenetic modification suppresses the FADS2 gene, which reduces the combustible fats in the membrane and stops ferroptosis.
Nailed it.
So, for the audience, consider this. We often think of genetics as fixed, right? And metabolism as this fluid shifting thing. But can you see now how the food a cell produces actively rewrites its genetic behavior?
It really challenges the traditional dogma of biology.
It totally does. Okay, we've covered so much ground today. Let's do a grand summary of this entire epic journey from start to finish.
Okay.
So the leukemic stem cell burns fat through FAO to fuel the enzyme HMGCS2,
which manufactures massive amounts of BHB.
That BHB then travels to the nucleus and alters the histones via KBSB,
which turns off the FADS2 gene.
And turning off FADS2 lowers the combustible PUFA fats in the cell membrane,
ultimately making the cancer cell completely bulletproof against ferroptosis.
It is a phenomenal chain of events. But, before we wrap up, I want to touch on something super provocative from the discussion section of the paper.
Oh, the ketogenic diet implications. We have to talk about this.
Right, because in the wellness space and even in some oncology circles, intermittent fasting and ketogenic diets are hugely popular.
Yeah, the logic is usually based on the Warburg effect, the idea that tumors love sugar. So if you go keto, you force your liver to pump out tons of systemic BHB, and you basically starve the cancer of glucose.
But wait, based on everything we just discussed, if these specific leukemic stem cells rely on BHB to suppress FADS2 and survive,
Yes.
could a ketogenic diet actually be providing these LSCs with the exact molecular armor they need to become invincible?
That is the paradox. The researchers explicitly noted this. In some different contexts, sure, ketogenic diets can suppress AML progression. But in contexts where these stem cells are the main drivers of relapse, flooding the system with exogenous BHB from a keto diet could actively protect the tumor.
It could literally be exacerbating the disease by shielding the stem cells from ferroptosis.
It's a stark reminder that cancer isn't a single monolithic entity. What starves one part of the tumor might perfectly optimize the survival of the stem cell hiding in the bunker.
Which brings me to the final question I want to leave you, the listener, with today. In the war against cancer, what if the quote-unquote healthy diet trend you are following is secretly feeding the enemy's armor factory?
It really makes you rethink personalized medicine, doesn't it?
It completely changes the paradigm. We can't just prescribe broad metabolic shifts without understanding the exact genetic and epigenetic makeup of the specific cells we are fighting.
Absolutely not.
Well, this has been an incredibly dense, fascinating journey. Thank you so much for breaking down this incredible paper with me.
It was a pleasure. Thanks for having me.
And to everyone listening, keep questioning the consensus, stay curious about the mechanisms operating beneath the surface, and thank you for joining us on this deep dive.